Hello, my name is Gemma Garcia. I'm a postdoctoral researcher on Jose Maria Millán's team. Our laboratory is located on the Health Research Institute of La Fe in Valencia, and we are also a member of the Cyber of Rare Diseases. Today, I'm going to talk to you about the genetic testing for Usher syndrome. Usher syndrome is a disorder of genetic origin and the pattern of inheritance is autosomal recessive. For each gene, we have two copies, one from the mother and one from the father. For the disease to be manifested, the two copies of a gene have to be altered, and these alterations are called mutations. So, the genetic testing for Usher syndrome must be able to detect the both mutations responsible for the disease. Traditionally, the molecular diagnosis for Usher syndrome has been performed analyzing gene by gene, starting with the gene with a higher prevalence in each clinical type. However, the high number of Usher genes, the large size of the genes, and the existence of atypical cases where the clinical features don't fit into an exact clinical type difficult the molecular diagnosis. Another strategy is the genotyping microarray. It's a rapid strategy but only able to detect mutations previously described. Several years ago, the complexity of diagnosing Usher syndrome has been solved by the next generation sequencing technology. This technology allows us to screen all the known Usher genes in several patients at the same time. We have developed a panel of genes including the 6 genes responsible for Usher syndrome type 1, the 3 genes related with Usher syndrome type 2, the 2 genes responsible for Usher syndrome type 3, and the 2 additional genes associated with the disease. Moreover, we have included also 4 regions in Usher2A gene where intronic mutations have been described. Briefly, the strategy consists in several steps. The first step is to obtain the material, the DNA, and we extract the DNA from blood or saliva from the patients. The next step is to cut all the genome in a high number of pieces. And then, using different systems, we are able to select only the fragments corresponding to the Usher genes, in white in the picture, and to remove the other ones, in gray. Then, we have to sequence to analyze all the small fragments corresponding to the Usher genes. The last step is to align, to map all small fragments to a reference genome. As example, imagine that the sentence, "When I saw you, I feel in love and you smile because you knew," is the genome of reference, the normal sequence of a gene. And the small fragments, the small pieces of the sentence, are the sequence we have obtained. With all the fragments, we are able to construct the original sentence, and comparing the fragments with the genome of reference, with the sentence, we are able to detect all the differences. For example, the A has changed to I, and this difference is called a variant. One of the problems we have to address is the interpretation of the variants. Why? Because when we analyze all the Usher genes, we detect more than 100 variants per patient. And we have to discriminate the benign variants from those having a pathogenic effect. For the assignment in polymorphic or pathogenic state, we rely in public databases. We also use bioinformatic tools to predict the effect of the variant. Usually we analyze also the relative, and sometimes additional functional studies are needed. Last deletions and duplications are not easy to detect using next-generation sequencing. For this reason, to detect this type of mutations, we use the CGH array, Comparative Genome Hybridization Array. We have the DNA from the patient, and we have the DNA from a healthy control. The patient DNA is marked in red and the control DNA in green. And then we have to compare the color intensities. If for a region our patient has an intensity lower than for the control, the patient has a fewer copies than the control, so the patient has a deletion in this region. We use next-generation sequencing since 2013, and we have analyzed more than 100 patients. In 89% of the patients, we have detected at least one mutation. In 71% of the cases, we have detected the both mutations responsible for the disease. In 18% of the patients, we have detected only one mutation, and in 11% mutations were not detected. In this picture, we can see the distribution of the mutations in the genes. We can see that in 43% of the mutations are located in ACER2A gene. The two other genes with a higher prevalence are MYO7A and GPR90A. Sequencing all the ACER genes allows us to detect the mutations in patients with clinical features, typically ACER syndrome type 2, having mutations in genes typically involved in ACER syndrome type 1, as MYO7A or cadherin23. As I say in the other slide, in 18% of the patients we detect only one mutation, and in 11% mutations were not detected. So the question is, why don't we detect all the mutations and in all the patients? So maybe the mutations are located in other genes associated to ACER syndrome but not yet described, or maybe these patients have other clinical features and they have other syndrome, not ACER syndrome, but with overlapping characteristics. Another possibility is that the mutations are located in non-coding regions. Why? Because the genes are composed by coding regions called exons and also by non-coding regions, in red in the picture. Usually for molecular diagnosis, we analyze only the coding regions, the exons in yellow, but not the non-coding regions. Why? Because usually the mutations are localized in the exons and because the non-coding regions are very, very large. So maybe some of our patients have mutations in these non-coding regions. We hope that these patients with only one mutation or with mutations will be solved analyzing the whole exome or the whole genome. It has been a pleasure to me to give this talk, and to end, I would like to thank to all the patients and the relatives for always collaborating in our projects, and also to all the associations for their help. Thank you very much.